Hello, class, and welcome to today's lecture on brain and behavior. I'm thrilled to have you here as we delve into the fascinating world of neuroscience and we go through that journey of discovering how the intricacies of the brain shape our behavior. And as we delve into the depths of neurons and cytoarchitecture and neurotransmitters, I want you to just reflect a little bit on how amazing you are and how the physical and the spiritual comes together to form your thoughts and emotions and behaviors. Now, as always, I like to start off with a little story, and today I want to just tell you a little bit and pay a little bit of homage to the pioneering researcher that really forms a large part of our foundational knowledge in neuroscience, Santiago Ramon Cajal. Cajal was born in 1852 and died in 1934 and is known as the father of modern neuroscience. He was a Spanish neuroscientist, and his groundbreaking work in the late 19th and early 20th centuries really laid the foundation for our current understanding of neurons and really the overall structure of the nervous system. Cajal's contributions to neuroscience really are profound. He was a pioneer in using a staining technique known as the Golgi method, which allowed him to visualize individual neurons under the microscope. And this breakthrough really led to his formulation of what we now know as the neuron doctrine, which states that the nervous system is comprised of discrete individual cells called neurons that communicate through specialized junctions known as synapses. And one of Cajal's most famous contributions was his detailed drawings of neurons and brain structures. His intricate and accurate illustrations not only captivated the scientific community, but also served as valuable educational tools for generations of neuroscientists to come. These drawings provided the first clear evidence of the structural complexity of the brain's neural networks. And Cajal's work was not only limited to the understanding of the structure of the neurons, he also made significant contributions to our understanding of neuroplasticity. That is, the brain's ability to adapt and rewire itself in response to experience and learning. His observations laid groundwork for later research in this area, which we'll explore as we go through this lecture. Cajal's dedication really to unraveling the mysteries of the brain has really left an indelible mark on the field of neuroscience, and his insights and discoveries continue to inspire and guide researchers in their quest to understanding the brain's intricate workings. So as we move forward in this course, just keep in mind the legacy of pioneers like Cajal, whose passion for discovery has fueled our understanding of neurons and cytoarchitecture and neurotransmitters. Now that we've paid some tribute to Santiago Cajal and his pioneering work into research, let's turn attention to the very building blocks of the nervous system, the neurons. Neurons are the cells responsible for transmitting information throughout the brain and the body, and understanding their diversity and functions is really crucial to comprehending how the brain works and how we behave. Now, the thing to keep in mind is that neurons come in various shapes and sizes and functions, and they can be classified into several distinct types based on their roles within the nervous system. So, I want to kind of start off by mentioning sensory neurons, also known as afferent neurons. And these are responsible for transmitting sensory information from our external environment or internal body to the central nervous system. And they play a crucial role in our ability to perceive and respond to stimuli like touch and taste and smell and sight and sound. For instance, when you touch a hot stove, sensory neurons in the skin send signals to your brain, promoting you to withdraw your hand very quickly in order to avoid injury. Then there are motor neurons, also known as efferent neurons, and they carry signals from the central nervous system to the muscles and glands, enabling us to execute movements and initiate physiological responses. So when you decide to move your hand away from a hot surface, motor neurons transmit the instructions from your brain to the muscles involved in that action. And then there are interneurons, also referred to as association neurons. And these are found entirely within the central nervous system, and they act as really intermediaries, relaying signals between the sensory and motor neurons. And these neurons really play a crucial role in processing and integrating information. And so it allows us to make sense of very complex sensory input and to generate appropriate motor responses. We also have mirror neurons, which are a specialized type of neurons that have been really kind of discovered more recently. And they're believed to play a role in things like empathy and imitation and understanding of actions and emotions of others. And when you watch someone perform an action, mirror neurons in your brain may activate as if you were performing the action itself, contributing to an ability to understand and connect with others. Another easy way to kind of think of it is if you're feeding an infant with a spoon and you're encouraging the infant to open up their mouth, you will often open up your mouth as your mirror neurons activate, and it's kind of a desire that theirs will activate as well, so they will open their mouths. And then there are pyramidal neurons and granule cells. So within the cerebral cortex we find pyramidal neurons, known because obviously of the pyramid-shaped cell bodies. And they also have very distinct complex branching dendrites. So they are involved in higher cognitive functions, including decision-making and conscious thought. And then there are granule cells, which on the other hand are smaller neurons found in regions like the cerebellum and olfactory bulb. And these seem to play critical roles in, first off, motor coordination as well as the processing of olfaction, that is, the sense of smell. So, as you can see, there are lots of different types of neurons, and understanding the diversity of neurons and their specialized functions is really essential because they form the intricate networks that underlie our thoughts and emotions and, of course, behaviors. Now, let's venture into the intriguing realm of neuroplasticity. Neuroplasticity really refers to the brain's remarkable ability to adapt, reorganize, and change throughout our lives. Now, first thing to kind of be mindful of, as far as the body goes, the brain really is something that does have difficulty healing itself, but to a limited extent it can do that. And so the fact that it's not purely 100% rigid is in itself remarkable. So neuroplasticity really is a concept that has revolutionized our understanding of brain function, and it has a profound implication for fields like psychology and medicine and rehabilitation. Now, a few key things to know about neuroplasticity. First off, we can think in terms of structural neuroplasticity, which involves physical changes in the brain's structure. And this can include the growth of new neurons, what we call neurogenesis. It can be the formation of new connections between neurons, which we call synaptogenesis, and the pruning of unused or unnecessary connections, which we call synaptic pruning. So one fascinating example of structural plasticity occurs during childhood and adolescence, when the brain undergoes significant changes in response to learning and experience. For instance, the brain regions responsible for language processing in bilingual individuals can exhibit increased gray matter density due to their linguistic experiences. Then we have functional neuroplasticity, and this relates to the brain's ability to reorganize its functions. If a particular brain region is damaged, other regions may compensate for its functions, and this phenomenon is often observed in cases of brain injury or stroke. For instance, if someone experiences damage to the motor cortex controlling the right hand, the brain may rewire itself over time, allowing the left motor cortex to assume some control over the right hand's movements. Now, it's still going to be a limited recovery, but any change is really fantastic. Then we have what is known as experience-dependent plasticity. And this really highlights the idea that our brains change in response to our experiences and interactions with the environment. And so learning a new skill, mastering a musical instrument, or even recovering from a sensory loss such as blindness or deafness can all result in significant neural reorganization. For instance, studies have shown that the brains of professional musicians have distinct structural and functional changes in regions responsible for motor control and auditory processing. And then we can also think in terms of critical periods, which are specific developmental windows during which the brain is highly sensitive to particular types of input or experiences. And these periods are critical for the establishment of essential skills and functions. So we can think in terms of a critical period in language development in children. The early years of life represent a crucial window for language acquisition. And if a child is not exposed to language during this time, it can have long-lasting effects on their linguistic abilities. So understanding neuroplasticity is essential because it challenges the traditional notions that the brain structure and functions are fixed in adulthood. Instead, it suggests that our brains may remain adaptable and responsive to change throughout our lives. And so this concept really kind of has far-ranging implications, from rehabilitation theories and therapies to brain injuries to enhancing cognitive abilities through training and education, and really a lot of the most fascinating research centers around neuroplasticity, because it is the place where we can not only understand what is going on, but we can affect change in terms of improving injury and improving the functioning and structure of a brain. And the more that we can do that, the more that we can help people with traumatic injuries to the brain, as well as those suffering from mental illness. Now, I want to kind of just delve into, as we kind of finish off this part of our lecture, into a little bit of the historical roots of cytoarchitecture. Now, if you recall, we looked at Cahill, and he really kind of had a lot to do with understanding cytoarchitecture. So we're going to delve into the historical development of cytoarchitecture, which is a field that has played a crucial role in our understanding of the brain structure and function. And really, cytoarchitecture, which roughly translated means cellular architecture, really focuses on the microscopic study of brain tissue to identify distinct cellular patterns and structures. And so this discipline has deep historical roots with some key milestones and figures that have shaped our knowledge of the brain's intricate organization. Now, if you recall, Cahill used the Golgi method. So we should probably explain what that is. Well, our journey really begins with Camillo Golgi, who was an Italian physician and scientist who in the late 19th century developed the Golgi stain, which was a revolutionary technique to visualize individual neurons. And so this stain allowed Golgi to observe and sketch the intricate branching patterns of neurons and brain tissue. And Golgi's work, however, was met with a great deal of skepticism. And so his belief that neurons were connected in a continuous network, which came to be known as the reticular theory, conflicted with the prevailing theory of the time, the neuron doctrine. And then we have, of course, Santiago Cahill, who made significant contributions to the field. So he used the Golgi stain to support the neuron doctrine, providing evidence that neurons are discrete individual cells with distinct structures. And so, as we've already mentioned, Cahill's meticulous drawings of neurons created through this Golgi staining method helped solidify our understanding of the brain's cellular architecture and ultimately discredited the reticular theory. Now, in the early 20th century, there was a fellow by the name of Franz Nissl who introduced another staining technique known as Nissl staining, of course, which highlighted cell bodies and allowed for a more detailed examination of neuron density and distribution. And so as we progress, we get these new types of tools to study. And the Nissl staining became really a valuable tool for identifying different brain regions and studying the effects of neurological diseases. And today, we have some fantastic advances in technology, such as the electron microscope and advanced imaging techniques, which have taken the study of cytoarchitecture really to new heights. And these tools allow researchers to study brain tissue really at the nanoscale, revealing even more intricate details about neuronal connections and structures. And understanding cytoarchitecture is really fundamental because it forms the basis for our knowledge of how different brain regions are organized and interconnected. And this knowledge, in turn, helps us comprehend how various brain regions contribute to specific functions and behaviors. So we're going to pause there, and when we come back, we will explore some fascinating areas of how cytoarchitecture kind of relates to brain function. So we'll see you in just a little while. I hope you can take a nice break, and when we come back, we will continue by taking a look at cytoarchitecture and localized brain function. See you in a few minutes. Bye-bye.